Hi, I'm Brad Power, and this is the Cancer Patient Lab. And today we're honored to have with us Dr. Ramesh Narayanan. He is a researcher in prostate cancer, and he will be talking to us today about what he's seeing in the drugs that he's been trying to bring to market, as well as, in general, what the landscape looks like in bringing new drugs to prostate cancer. And I'm sure we'll have a stimulating conversation about just understanding what the state of the art is and what patients should be looking forward to in the months and years upcoming. So with that, I'll turn it over to Dr. Narayanan.
Brad, thank you so much for having me here. It's an honor to meet you all, and it's a privilege to talk to you. Um, I've been thinking in the last 5 minutes how to condense a 30-minute presentation into 10 minutes, but let me just go over a few things that would be helpful here for setting up for a discussion. As Brad pointed out, we have our group has discovered drugs for different diseases, and the lead drug that is in the phase one clinical trial is for treatment refractory and relapse castration-resistant prostate cancer. We received FDA's fast track approval for our drug, and we are in the process of raising about $25 million to advance it to phase 1B and phase 2 clinical trial. We co-founded this company called Ramiller, combining my name Ramesh and Dwayne Miller. Miller is our co-investigator. He is the lead chemist in the program. So we created this company so as to advance our drug into the phase 1B clinical trial. Okay. So I just want to give you one background, one topic that keeps coming up every single time. Probably you all take a medicine or something is androgen and androgen receptor. So how did androgen and androgen receptor became the driver of, or how was it discovered that they are the drivers of this prostate cancer? So what I'm showing you here is the Nobel Prize winning discovery by Huggins and Hodges. What they did was our prostate, which for normal development of prostate requires androgens. So that was well known in the 1930s and 1920s. And what they thought was that if the normal prostate development requires androgens, probably the same androgens are contributing to the development of the prostate cancer. So what they did was they performed two clinical trials. Of course, in those days they were not controlled clinical trials, and one of them was surgical castration. So they surgically eliminated the testes, which is the primary source of androgen synthesis in our body, and then they wanted to see what happens to the cancer. And those days there are no biopsies, there are no other diagnostic available. So the only way to detect whether a cancer, prostate cancer, is shrinking or not is using an acid phosphatase colorimetric assay in the blood. So they were able to show that by surgical castration, that by cutting off the testicular source of androgen supply, they were able to shrink the prostate cancer here. And androgens bind to the receptor called androgen receptor. That's why either cutting off the androgen supply or blocking androgen receptor, which is the current modality of treatment, have really helped us to advance the discoveries based purely on Huggins and Hodges. The other thing that what they did also was that administering estrogens are female hormones, are women hormones, and what happened was that when you administered estrogens, it cuts off the testicular supply of androgens based on this panel that I'm showing on the right. This is called hypothalamus, pituitary, and gonadal axis. So our hypothalamus synthesizes gonadotropin release hormone that stimulates the pituitary to release luteinizing hormone and follicle stimulating hormone. And finally, this releases testosterone. By like tricking the brain that there is already a surge in the hormones, the brain shuts down the synthesis of testosterone. And this is the second modality by which they were able to show that the tumor regresses. So these set up the future discoveries of androgen receptor antagonists and everything that we are currently taking and which are in the market, which are pretty much the mainstay treatment options for our for prostate cancer. So let's just leave some of these background slides, which you all have seen, or which are quite basic information, which we don't have at this point. So this slide, what it shows is that androgens, they come into the cell and bind to the androgen receptor. So the androgen receptor, which is in a conformation, it just changes the shape and then it binds to the DNA and then causes the transcription and translation of genes, including our PSA, which has become the primary diagnostic marker for prostate cancer. So either you inhibit the steroid entry into the cells or completely inhibit the steroid synthesis, which is what abiraterone does, or inhibit the androgen receptor, so that which is what enzalutamide, apalutamide, darolutamide, like Xandy, all these drugs they are all doing. By either way, you're blocking this particular receptor. So this receptor is present in over 80 to 85% of the prostate cancer patients, and remaining 10 to 15% of the prostate cancer do not express the receptor. So, which means that predominantly majority of the prostate cancer will respond to androgens or androgen receptor blockers. So, for example, if you take here, the prostate cancer evolves from hormone sensitive prostate cancer, an early stage prostate cancer, which is contained within the prostate. And the primarily prostatectomy or radiation or gonadotropin releasing hormone, which I showed you before. These are the primary treatment options for this stage. When it becomes hormone sensitive and resistant prostate cancer, then you have several options like androgen receptor blockers like enzalutamide, abiraterone, darolutamide, apalutamide. Then it becomes non-metastatic castration-resistant prostate cancer. Again, androgen receptor antagonists are the primary source of treatment here. Then it becomes metastatic castration-resistant prostate cancer, which is a very difficult disease to treat and manage. And then again, you have pretty much the second generation androgen receptor antagonist, chemotherapy, lutetium, PSMA, and PARP inhibitors are there. And then finally, it's unfortunately palliative care. So the prostate cancer number of patients in globally is just massive right now. It's about annually 1.6 million men are diagnosed with prostate cancer, but this number is likely to expand to about 2.5 million people, men, and it's going to contribute to about 700,000 deaths annually from by 2040. So how do we manage this prostate cancer that's evolving every single time? So what happens is that I just want to go to one particular slide here. So if you look here, every single time you block the androgen receptor, cancer is very smart. It's always going to find an escape route. So escape routes are different escape routes, like androgen receptor becomes sensitive to other hormones or splice variant, which will go through what it is, and different kinases and different ways. These are the different mechanisms by which androgen receptor finds escape routes. So every single cancer, not only prostate cancer, but every cancer always tries to rebel against the blockers. So if you look at this slide, this is the androgen receptor structure. It is a classic transcription factor structure. It has an N-terminus domain, DNA binding domain, hinge region, and the ligand binding domain. All the drugs that are being developed right now bind to this domain and then block this androgen receptor from functioning. Cancer being very smart, what it does is that it just cleaves off this particular domain and creates this constitutively active monster. This one retains about 75-80% of the androgen receptor activity. And now you don't have the ligand binding domain here for it to further inhibit the prostate cancer. So what happens is that this becomes the driver of the cancer. And the patients who express the splice variants succumb to the disease very quickly in about a year or year and a half. So it's a very aggressive form of prostate cancer. Right now, the challenge here in the prostate cancer drug discovery and development field is that we all know how to block this one, but can we block this? This is where our drug comes into play, where our drug binds to this region. So by binding to this region, not only we are inhibiting the full-length androgen receptor, we are also inhibiting this spliced variant androgen receptor. Thereby, we are able to take down both the full-length and the spliced variant forms, and the prostate cancer just shrinks or gets contained by our drugs. Okay, so what are the current therapeutic landscape of prostate cancer? As I mentioned, the localized or hormone-sensitive prostate cancer, you have radical prostatectomy, brachytherapy, there is radiation, and some of these other treatments like enzalutamide, apalutamide, and darolutamide, which all showed significant enhancement in the metastasis-free survival as well as in the overall survival. So overall, this is really a good thing. But of course, As you keep treating the prostate cancer, as you keep hitting the androgen receptor, they start creating new forms or new mechanisms by which they can overcome the inhibition. So this is hormone-sensitive prostate cancer. Again, you can see that both apalutamide and darolutamide are approved for this one, and you can see that they are pretty much they can extend the progression-free survival or even overall survival significantly in this patient population. This is the major issue. That is the metastatic prostate castration-resistant prostate cancer is the major problem right now we have, how to manage it. Right now, you have enzalutamide and abiraterone are approved, and I think darolutamide and apalutamide are also approved in this case, where you can see that they performed the studies in chemo relapse and chemo naive patients. That is, those who have relapsed from the chemotherapy, as well as those patients who have never experienced or never been treated with chemotherapy. In both cases, the overall survival improved, like say 18.4 months as opposed to 13.4 months, a marginal increase, but still it's all statistically significant, like about 4 to 5 months on an average, statistically significant. So AR antagonists and androgen synthesizing inhibitors, they can actually provide a good amount of relief or an extension. And beyond this point is where we are all trying to discover drugs and develop drugs so that people or men who have relapsed from these cancers, can they be put on new modality of treatments, and that can actually pretty much give them more runway before they go to chemotherapy. This is what all our approaches are. Of course, you have chemotherapy. You all have would have experienced, like some of you would have experienced docetaxel or cabazitaxel. We, as much as possible, we want to avoid chemotherapy because these are all nasty drugs. They don't differentiate or discriminate between the cancer and the normal cells. So we don't want the patients to go through any of the side effects by taking on chemotherapy. So we would love to have provide additional hormonal treatments that can actually give them longevity in terms of the targeted therapeutics. Then you have radiopharmaceuticals are coming along really well. PSMA is a membrane antigen in prostate cancer. And this is something that you can just develop an antibody and combine that with lutetium and then focus that onto the cancer. So this is something that is being now developed by many companies, and they're showing really good success. And you have PARP inhibitors, which are exclusive for BRCA mutations. We were initially thinking that BRCA mutations are exclusive to breast cancer and ovarian cancer, but now a small subset of prostate cancer patients also express BRCA mutations. And these patients are really very good candidates for going on to PARP inhibitors, which can provide really a nice runway before you go to chemotherapy. So what are the therapies right now in development? You have several drugs that are going on in development. For example, PROTAC is a new modality of treatment. There's something called RIPTAC. As we don't have much time right now to go into details of this, many of these are right now in the clinical trials, and everyone is trying different approaches to see if they can provide additional benefits to the patients who relapse from existing treatments. So this is something that is all coming up. Recently, in 2 days ago in ASCO, a Chinese company presented another PROTAC that is showing really a very good response. There is a RIPTAC is something that actually brings in the androgen receptor with the BRD4 and inhibit that, and that was providing a 70% PSA 50 response. So what does our drug do? So our drug actually, this is the androgen receptor. This is just a modeling that I'm showing. Our drug binds to the disordered N-terminus domain and changes the conformation of the N-terminus domain of the androgen receptor, brings in a E3 ubiquitin ligase, and then chews up the androgen receptor and the splice variants. This is the mechanism. So when we performed the phase 1A clinical trial, we actually had 20 patients, CRPC, who were heavily pretreated. Average treatment was about 3 to 4 different treatment options where treatments the patients have gone through. And 6 patients were still on study when we discontinued the study because of financial reasons by the previous company. And then it came back to the university and I have licensed it out. 11 patients completed, of whom 5 patients showed really a good response in PSA, anywhere between 25 and 50% decrease in the PSA from baseline. More than that, what we were also able to show was that the androgen receptor was degraded in 7 out of 10 patients within 28 days, which means that we are not only blocking the androgen receptor, but we are actually getting rid of the entire androgen receptor from the system so that the patients will respond far better over a prolonged period of time. So two patients had stable disease in the BID dosing. There's twice daily dosing and a 16% decrease in tumor burden within 28 days. So these are all very encouraging signs with our drug. And we are hoping that within the next 2 to 3 months, we are very aggressively raising capital for advancing this drug, put this back into the clinical trial so that the patients can be benefited. And we can make sure that our science has gone out and benefited the patient community, prostate cancer patient community. So where we started in 1941 was the surgical castration, and we evolved tremendously over the last 80, 85 years. And today you have several treatment options. Yet the prostate cancer is pretty much contributing to a significant number of deaths annually. Within the USA, about 30 to 35,000 men are dying from prostate cancer. And we hope these new modalities of treatments will provide additional survival benefits to the patients so that the ultimate objective for researchers like me is to make prostate cancer a chronic disease rather than a death sentence at some point. So we are just striving to find new drugs and new modalities to make sure that we can just make the prostate cancer a chronic disease like diabetes or cholesterolemia. With that, I'll stop and then we'll just open it up for really good discussion here. Thank you so much.
Thank you. That was a good job of streamlining it down to 15 minutes on the fly, and provided an excellent summary, I think, of the state of the art. As usual for everyone here, if you have questions or comments, you can use the raise hand feature, or you can submit them through the chat function, and I'll read them to Ramesh. Anybody with any questions off the top here? I know, Russ, I would expect you might have some questions or comments if you can.
Yeah, sure. Is there data to suggest that PARP inhibitors might be effective if you're not BRCA mutated or HR mutated?
That is what right now the thought is, but people are even starting to try PARP inhibitors, even in those who have wild type BRCA. And some preclinical evidence are suggesting that it's true, but it's a big leap of faith that you have to take when you go into clinical trials, whether you can put the PARP inhibitors into the one of the biggest a discouraging thing for prostate cancer, like breast cancer and ovarian cancer, is that the immunotherapy is not quite effective because these cancers are cold cancers. There is not much immune infiltration. So, is there any way that you can prime the pump for immune infiltration so that the immunotherapy can become successful? If that can be done, then it will really be very helpful for prostate cancer patients to have additional line of treatment for that. So, but to answer your question, BRCA mutants are the ones currently being, those patients are the ones approved with the PARP inhibitors. But there are studies that I have come across where people are trying to see if it can be extended to non-BRCA mutated patients also.
Okay. Are you aware of Saraparib? I'm not sure if I'm pronouncing it right. Saraparib. Are there other PARP1 selective inhibitors in trials
So,
now?
the different PARP inhibitors are like say olaparib, rucaparib, and talazoparib. These are the three ones that are approved right now. If I'm right, you mentioned cereblon, right?
Saraparib. Saru Parib.
Cereparib, I have not. I'm not familiar. I think only the three these rucaparib, talazoparib, and
Those are the
olaparib are the approved ones for prostate
Yeah,
cancer.
those are the three approved ones.
Yeah.
Saraparib is in, I think it's phase 3 clinical trials now.
Hmm.
It's showing some activity in HSBC and HRR wild type.
Hmm.
So no mutations whatsoever.
That
But again,
that that
it's in phase 1 and phase 2, and I believe it's in phase 3 right now. Pretty sure that's right. It's in phase 3 now, and I was just wondering if there are other ones. I'd say a PARP1 selective inhibitor. So only instead of PARP1, PARP2, it just inhibits PARP1, and maybe that has something to do with it. It's being things working for HRR wild type?
No,
Not sure.
so I'm involved with a company that's developing a PARP1 selective inhibitor for
Okay,
certain
that's what
cancer.
that is. That's what it is.
And the primary reason for them to have a PARP1 selective inhibitor is to reduce the side effects, like
Oh,
hematological
effects.
side
Mhm.
effects and other things, which are
Thank you, David.
evidently attributed to PARP2. So that is the reason that they are going after PARP1 selectivity. But it would be a transformational outcome if they are able to show a statistically significant outcome in PARP in BRCA wild type patients with new PARP inhibitors. That would really be very helpful.
Um, is anything else, Russ?
That's actually what I was interested in is the
Okay.
PARP inhibitor part of it.
Okay. Let me
Um,
ask. Oh, I was going to just ask David Plunkett if I could put you on the spot. David, I'm just curious since I know you're very educated on all of the treatment options that Ramesh went through. Was there anything that you learned or anything different? Just if you could give me sort of an editorial comment on what you learned today.
What I was seeing there looked very familiar. It was a nice recap, I think.
Mhm.
I'm interested to hear or rehear the name of the drug that you're currently working on and ask, is that still in a phase 1 trial, or is the phase 1 trial extended, or are you ready for phase 2?
Mhm.
So we went our drug earlier was called ONCT 534, was run by Oncternal Therapeutics. So it was discovered in our lab at the University of Tennessee. So it came back to the University of Tennessee after Oncternal shut down the company because of financial reasons. So then I decided to take the bull by its horn. So we took the drug and then we are starting to develop. So right now, the IND is it's a fast track IND. The IND is transferred back to us. So it's an inactive IND. The minute we develop, so we need about $6 million to get to the phase 1B. Phase 1A was run between 40 milligrams and 1,200 milligrams and 300 milligram twice daily. So we found significant activity at 300 milligram twice daily. So what we are now doing is that we are going to narrow down to 18 patients to treat them with 300 milligram twice daily and 450 milligram twice daily to just make sure that we choose one of the doses to go into phase 2. So we are likely to start the manufacturing. Everything is ready. The minute we get the funds into our account, we will just start the manufacturing of the API as well as the drug product, and then initiate. Ideally, we would like to initiate the trial by the third quarter of this year, and then the phase 2 trial by 2027, third to fourth quarter. This is the timeline, right? We have, and we are working very hard to just make sure that this is becoming available to the patients as fast as possible.
What are your thoughts on the qualities of the patients you want to recruit? What
So
characteristics will you be looking for?
So we are looking for patients who have already been treated with multiple lines of treatment. So the phase 1A trial, the patients were treated all the way from different AR antagonists, androgen synthesis inhibitors, and also chemotherapy. And then we recruited them so as to make sure that we start late stage. And then if we are showing good success, then we'll just move early stage because some of these current AR antagonists, they have some liabilities like GABA inhibition, which causes seizures, and we want to avoid those, and ours do not have that. So we are hoping that we can just start at late stage, which is easier for approval, and then go early into the different patient population.
On the other side of the coin, are there any characteristics that would disqualify for a trial?
So we have not come across. So overall, the phase 1A trial was extremely safe. In fact, it was only at the highest dose, the maximum tolerated dose was achieved at 1200 milligram, where they saw a typical AR targeted side effects such as hematological problems. But once the patient came off that dose, the patient reversed back to healthy condition. So we, at the dose that we are proposing to treat, we found that it was quite safe. There were no side effects. Maximum grade 1 or 2 toxicity is just some nausea, vomiting, but we don't know whether these are associated with the drug or the condition of the disease. So we are not sure. But as of now, I can say that the drug is extremely safe.
Is that okay for you, David? Yeah. Let me just ask some of the other prostate cancer patients on this webinar the same question I asked David. Alexander, I know Chad, I think Arthur and Chase are also prostate cancer patients. Anybody want to comment on the summary? I think that Ramesh did a very good summary of the treatment options available and where they're fitting and so on. Anybody have any thoughts or comments on that? Alexander?
I just wanted to ask, what is the efficacy tested, and what is the efficacy on resistant prostate cancer patients?
Of which treatment?
Of our drug?
The R.
Okay. So the
ones
efficacy
that are still hormone sensitive, but the tumor continues to grow.
so the till the end stage, pretty much almost all the prostate cancers, they can be treated with hormonal treatments because the androgen receptor, one form or the other, remains active at that point. But what happens is that when you keep hitting the androgen receptor, they become mutated, and the mutation will not permit the antagonist or the inhibitor to work further. So that's why different people are trying different approaches to block that androgen receptor, the mutated or the transformed androgen receptor. So this is pretty much about 85%, 80 to 85% of the patients do still respond to androgen targeted treatments.
And follow up, if the patient is treated with enzalutamide, for example, after the enzalutamide, if I understand right, these drugs are less effective, or
Exactly. So, for instance, when you're treated with enzalutamide and then you relapse out of it, they try to go for, like, say, darolutamide or apalutamide, but as you rightly said, the cross resistance is another major problem. So targeting the same androgen receptor. So this is why people are trying different approaches. Like, for example, in our case, we bind to a domain that is completely different from what typically everyone binds to. So by that way, we are not tapping into the same region where all the drugs are shown to have cross resistance. We are going to a completely different domain, which we all in scientific terms call as non-canonical domain, and then try to block the androgen receptor. By this way, the cross resistance does not take place. And we have other mechanisms also where we have evaluated in models which are shown to be pan resistant to all the inhibitors, and we still show efficacy in those. So considering these are quite encouraging preclinical data. And also the clinical trial, the patients were treated heavily with enzalutamide or apalutamide or one of these other amides. And we are able to show an inhibition in the PSA of the patient. So the patients, when they were recruited, 2 or 3 continuous monitoring, their PSA was increasing. But when we put them on our drug, the PSA started coming down within like 4 weeks, which shows that the drug is pretty much working the way that we expect it to work.
Okay, Alexander? Okay. There's a question in the chat from Alan Morris. He says, I know this is off topic, but was wondering if you could weigh in on the concept of cold tumors. You must make it hot first. So he's talking about immunotherapies in prostate cancer, I guess. Just generally, what are your? I don't think you touched much on immunotherapies. Could you just give your perspective on that?
Yeah, it's very unfortunate that immunotherapy fails, or rather failed in prostate cancer, typically similar to breast cancer or ovarian cancer, one of these hormonal cancers. So one concept that is out there is that these tumors do not have immune infiltration. So that only when the T cells are infiltrating into the tumor cells, you can use these PD-1, PD-L1 inhibitors that actually create this environment to block the tumors. Like melanoma is a great example, lung cancer, even bladder cancer seems to be quite responsive because last week there was a clinical trial that came out suggesting that the pembrolizumab was quite effective in bladder cancer. So this is all quite effective. But when it comes to prostate cancer, we have been quite struggling with that. So we are in our lab have developed another set of molecules or drugs, not drugs, but it's a molecule where we are creating stress in the cells, cancer cells. That stress creation, we think that it could potentially have a changed microenvironment that will bring the T cells into it, and then we can combine it with the immunotherapy. It's a hope right now, but there are a lot of other people are also trying this approach of how to just tap into more immune system in the prostate cancer microenvironment so that we can treat it with the immunotherapy. The clinical trials have not been quite successful, but we are expecting it to just have some kind of an effect quite soon.
I'm trying to remember the name, but I believe there is one approved immunotherapy in prostate cancer. It begins with an S, something like
It was Dendreon as Provenge was the one that was the first drug that was approved way back in 2007, 2008. The problem, in prostate cancer, patients would love to have oral treatment rather than an infusion. And that was regarded as one of the causes for Dendreon to go out of business and was not able to be successful in terms of advancing. In fact, that was the first immunotherapy, in fact, approved for any cancer. In fact, then only came the PD-1, PD-L1 and everything. But this is something that we have to keep our eyes and ears open for future immunotherapy.
Okay. And Dr. Apfel, if you don't mind my asking, is there a possibility for functional testing related to prostate cancer? Because many people are getting a prostatectomy, so there's tissue. You can have fresh tissue, which is the key ingredient. What has the experience been, or what questions would you have around using functional testing to predict which of the variety of possible therapies would be useful for different patients?
Yeah, that's an important question. Thank you for asking. I first wanted to comment on Dendreon because I thought this was actually an interesting case because the clinical study actually showed significant effectiveness with very little side effects. And the reason why it actually failed commercially was kind of because of costs. And so it was then, I think, sold to a Chinese. And so a Chinese company acquired them. And I think they were available in the Asian and Pacific area. And I'm not sure whether they are back here in the United States available. And many oncologists says, oh, the trials, the effectiveness was not that great. But if you actually look at the effectiveness, there was a lot of crossover in the trial design. And so if you take out the crossover, the effectiveness was actually pretty good. I think it was somewhere in the range of 20%. Overall, that's kind of now my rare recollection. So I still think the Dendreon is actually a very, very interesting solution if it works, of course. And of course, many of those tumors are not that hot, but I do think it is something that has a favorable risk-benefit profile, I would like to say, for those where it works. So I think this is a great one. Now, coming back to your question on functional profiling, our assay is particularly helpful when the tumor is relatively aggressive, so when it's a high-grade tumor, and when the tissue that we have has a significant tumor proportion. So if it's, let's say, a core needle biopsy from a prostate cancer, then you often have a lot of benign material, and that can confound those results. In a prostatectomy, it really depends on whether this is really tumor tissue. And so what we feel is the cleanest approach is if there is a metastatic stage, that we get actually a specimen from a prostate cancer metastasis. And we have been able to, even with small amounts of tissue, to isolate prostate cancer from even bone metastases, which is more difficult than isolating tissue from breast cancer metastases, because the bone metastases tend to be more osteoclastic, so they build more bone material around it reactively, as opposed to the breast cancers that are actually much easier to process. But we actually can process the prostate cancer metastasis from the liver, the lung, the bone, wherever it comes from. And that's probably the better approach than directly getting it from the prostatectomy.
Ramesh, do you have any comments on that?
No, this is great.
Right.
Actually, while Dr. Apfel was talking, I was just thinking about one of our publications. The investigators are KOLs from University of Wisconsin who were part of the clinical trial. They developed a very nice companion diagnostic from circulating tumor cells, and they published it about 4 or 5 weeks ago. And you all can search for with a keyword in PubMed, ONCT 534, ONCT-534, and they provided some really good biomarker things. So I was just thinking, probably Dr. Apfel and I should correspond after this talk at some time to just see what best we could do to develop some companion diagnostics for our phase 2, phase 3 clinical trials.
Happy to connect.
Yeah,
I can put down my email in the chat.
That would be great.
From Sophia Wren, there's a question about personalized vaccines, I guess mRNA vaccines, and wondering whether they might be promising in prostate cancer. They've been successful in other cancers. Have you got an opinion on that?
To my knowledge, I have not seen any activity on that in prostate cancer space, in fact. So I'll have to go back and check what is that. At least in the clinical development, I have not seen an mRNA vaccine. I can let me just go back and check my memory. There are some there's one gene therapy that is actually going on right now with some viral-based gene therapy, but mRNA vaccine I've not paid any attention, or I've not seen much in the clinical trial at this point. So
Okay.
it's an interesting question.
All right. Alan Morris has his hand raised. Alan?
Hi, doctor, and I cannot pronounce your name. I'll just call you Dr. N. Is that okay?
That's perfectly fine. Or even Ramesh would be better.
Yeah. I have to warn you that I'm very focused on the immune aspect of prostate cancer care. Thank you, Chris, for telling more about how Dendreon first went bankrupt and what the reasons were, and that now a Chinese firm has bought it out. So it still exists. The questions I'm going to ask you, because you are an expert, you're a scientist, you're at University of Tennessee, you swim in the world of people that are researching this. I want to explore the conventional wisdom with you. I think it's already been implied, but I want you to overtly state it. The conventional wisdom is that hot tumor, as opposed to the cold tumor, is the best tumor for a vaccine. Is that not the conventional wisdom?
Yes.
Okay, thank you for confirming that. Let me see. The corollary of that is that a cold tumor, and the prototype of that is prostate cancer, and that would be the worst tumor to pursue a prostate cancer vaccine for. That is the conventional wisdom, right?
Yes. Unless, yeah.
Yeah, you are actually doing important work for me, and I want to ask you if you will allow me to cite your hand on the pulse. I'm writing a paper. It's now book length, and it's why prostate cancer is the best cancer to pursue a cancer vaccine for. And you realize by me just stating that, I am, if you are correct in your hand on the pulse, I am stating something that is absolutely not believed in the medical and scientific community. Is that it? Would that be a fair statement?
I cannot say that absolutely not believed, but actually, still we think there is a hope for a vaccine in prostate cancer for sure.
Right.
Because as Dr. Apfel pointed out, that Provenge was a good success, but for varying reasons, it did not become a commercial success. But I'm sure that there is an opportunity to get into, but who is interested in investing large amount of resources to take that risk is the biggest question right now.
But no, I agree. And if I may probe you psychologically, you just demonstrated the measured response that all scientists take. They don't make absolute statements. And I admit I made absolute statements. And for example, if I write a paper, I should not make absolute statements, especially if I'm proposing something that's not conventional wisdom. May I want to, now that we've stated that the conventional wisdom is that the hot tumor is the best tumor to pursue a cancer vaccine, and that the cold tumor is the worst. The conventional wisdom also is one of the reasons it's the worst is because you first must make a cold tumor hot. In other words, there's this first pre-step that you have to do in order. So obviously, if we don't even know much about vaccine development, if we first have to do this Herculean step of making a cold tumor hot, then it should be that much more difficult to make a cancer vaccine to prostate cancer. Would that be in line with conventional wisdom?
It's the point here is that it need not be a Herculean task. There could be an opportunity with some modality that is under development or that has been developed that could be combined with the immunotherapy to just make the cold tumor hot. But what it is something that not many people know, or maybe people are evaluating it right now. But it's something that would be it need not be a huge battle, but it could be something that if people find it out, then it would become the mainstream treatment option.
Alan,
Okay.
Alan, let me interrupt. Knowing that you are sort of leading the witness through a long argument, could you just cut to a single question, please?
That is difficult. So the single question is, would you be willing to read my 50,000-word treatise on why prostate cancer is the best cancer to pursue a cancer vaccine? And what if you were willing to even attempt to read just the introduction and abstract and prologue, I would be forever grateful. Would you be able to provide me your email address?
Sure, I would be happy to read it, and it will be informative for me too. For everyone, for you and everyone, I'm just putting my email address here.
May I make one other comment? I used another absolute term, which is Herculean, and you used your scientific measured half to say no, it may not be Herculean. May I just synopsize it and tell me if it's correct? The leap is over the tumor microenvironment. In other words, if you believe in, like Boston Gene believes, that there's only an immune excluded state and not an immune naive state, you must. The exclusion is by the tumor microenvironment. Obviously, the cancer cell would be theoretically in there, but a major component of this is the tumor microenvironment. And you do know that everybody is researching the tumor microenvironment and how to modulate that. So that is the extra steps. And testament to how my Herculean word is not particularly exaggerated is they've been trying to do this for a decade and a half, if not more, and have been unsuccessful to date. And you mentioned the counter argument. Well, there's no money in it, and I in some sense I agree with that, but I don't agree with that. Basic science is pursuing this, and the tumor microenvironment is being dissected, but it's in its infancy. And I know your main talk is on AR degrading. Actually, I didn't quite understand your because I was focused on my own stuff. Is it mainly an AR degrading, a binding of the constituently activated AR state that you might get in? Is that what your molecule is doing, or
no?
Yeah, go ahead. Explain it to me.
Our molecule binds to a common domain that is expressed in this splice variant that is constitutively active, as well as in the full-length receptor. So it can inhibit all forms of androgen receptors. So that is the best part about this particular molecule.
But its main mechanism then would be to thwart the permanent on state of the constituently activated variant. Is that not right?
Yeah, so it is going to inhibit or degrade the whole AR constitutively active one as well as the full length receptor.
And to also summarize it, your company, you have a very promising thing, but you need the money to get the thing going as far as getting a real study to show to make it practice changing.
Yes, already the phase 1 clinical trial showed that it's a quite an effective drug. Now we need to advance it to phase 1B, phase 2, and phase 3 to just make it as quickly available as possible to the patients.
You may have said this in your talk, but you have people that are competing with you in the race. Can you list 2 or 3 that are competing in this, trying to molecularly thwart the constituently activated AR state? Who are the 2 or 3 that are competing with you, and where are they?
No one exactly in this space. So we are the only ones who are binding to this region and degrading the constitutively active form. So from that perspective, there is no competition at all. The remaining competitions come from the typical ligand binding domain binding inhibitors or degraders or some other modalities like gene therapy or so. But what we are developing is extremely unique, and this is why FDA gave us the fast track.
Okay,
Congratulations to you.
Oh, thank
thanks.
you. Thank you.
Ramesh, I always like to give an opportunity to the discussion leader, you in this case, to give some sort of parting words of wisdom or thoughts, key messages you'd like to leave with us.
Sure. Yeah. So I have put my email address in the chat. Anyone has any questions regarding the drug development or a mechanism of action or anything, feel free to reach out to me. That is one thing. Second is that there is a lot of hope in prostate cancer because this is something that every pharma wants to have a drug developed or every company wants to develop. The reason being that there is a large population of patients who are affected by this disease. And we researchers, like say in academic realm, as well as in many of the commercial side, they don't want to see the patients go on to chemotherapy. So what is the best option that we can give? And since this one has been studied, the androgen receptor and the whole prostate cancer mechanism have been studied for a sustained period of time, people are coming up with new approaches to target this disease. So what I'm seeing is that it's quite encouraging. Over the next 5 to 10 years, we all may have additional modalities, hopefully including ours, that can actually provide a sequential treatment for prostate cancer, like how breast cancer is currently being managed by multiple treatment options. The same thing, prostate cancer will have different options. And I'm very confident that the road is quite very promising right now. And I hope I'll be one of those who make a change in this to take care of the patients. Thank you all. Really, it's an honor to be here. And I just thank Brad for providing this a forum to talk to you all. And I would be very happy to come back and talk about any specific topics that you all are interested in the future.